Have you ever paused, perhaps in the quiet moments before sleep, to wonder what you are truly made of? Not just the muscles and bone that carry you through the day, but the very essence that lies beneath the surface of your skin. If we were to peel back the layers of our biological reality beyond the cells and the tissues, we would eventually find ourselves standing at the precipice of a silent, invisible world, a world where the laws of chemistry dictate the dance of life itself. Tonight we begin our exploration here, at the foundation of all things, as we seek to understand how a handful of simple elements from the Earth's crust and the stars above can come together to create a thinking, feeling, and dreaming human being. This is our first step into the chemical basis of life, a journey that starts with the humble but profound building blocks we call elements, atoms, and compounds. Lesson 1. Elements, atoms, and compounds. In this first session, we must look at what I like to call the alphabet of the universe. Just as a poet uses a few dozen letters to create an infinite number of verses, life uses a very small selection of ingredients to build everything from the smallest bacteria to the vastness of a redwood tree. We are moving from the macro world into the micro, peeling back the curtain on the substances that cannot be broken down any further without losing their identity. We will explore why you are primarily made of just 4 specific ingredients, and how the structural arrangement of a single atom determines whether it will become a gas in the air or a solid part of your skeleton. Take a deep, slow breath and let the complexity of the day fade away, replaced by the simple, elegant logic of the elements. When we examine the physical world, we find that it is composed of matter, anything that occupies space and has mass. Matter, in all its diverse forms, is made up of elements. An element is a pure substance that cannot be broken down into other substances by ordinary chemical reactions. Imagine an element as a pure note played on a single instrument. It is fundamental and unchanging in its essence. While there are 92 naturally occurring elements on the periodic table, the vast majority of life is constructed from a surprisingly narrow palette. In fact, if we were to look at your own body, nearly 96% of your mass is comprised of just 4 elements: oxygen, carbon, hydrogen, and nitrogen. These are the big 4, the primary colors of the biological world. To understand these elements, we must look at their smallest functional units, the atoms. An atom is the smallest unit of matter that still retains the properties of an element. I want you to try visualizing this for a moment. Though we often think of atoms as solid little balls, they are actually mostly empty space, vast silent cathedrals of energy. At the heart of this cathedral sits the nucleus, the atom's heavy central core. Inside the nucleus, we find 2 types of subatomic particles that provide the atom with its mass. First, there are the protons, which carry a positive electrical charge. The number of protons is the identity card of the atom. It determines which element the atom belongs to. If an atom has 6 protons, it is always carbon. If it has 8, it is always oxygen. Alongside the protons, we find neutrons, which are neutral and carry no charge, acting as a stabilizing force that helps hold the nucleus together. Swirling around this dense center in a cloud of constant motion are the electrons. These are much smaller than protons or neutrons and carry a negative electrical charge. Because they move so quickly, we do not think of them as being in one fixed spot. but rather as existing in shells or energy levels around the nucleus. It is the attraction between the positive protons in the center and the negative electrons on the outside that keeps the atom from falling apart. It is a perfect, vibrating balance of pull and push. When these atoms do not wish to remain alone, they find partners. When two or more different elements come together in a fixed ratio, they form a compound. This is one of the most beautiful transitions in chemistry, for when elements join to form a compound, they develop what we call emergent properties. This means the compound has characteristics that the individual elements did not have on their own. You might picture this as a transition from a single note to a complex chord. For example, sodium is a highly reactive metal that can explode if it touches water, and chlorine is a poisonous gas. Yet, when they bond in a 1-to-1 ratio, they form sodium chloride, common table salt, which is not only harmless but essential for your nerves to fire and your muscles to move. The compound is entirely different from the ingredients that made it, showcasing the transformative power of chemical organization. As we rest with these ideas, we can begin to see that nature is not just a collection of parts, but a master of organization. To understand why certain elements behave the way they do, we must look at the specific numbers that define them. Every element on the periodic table is assigned an atomic number, which is simply the count of protons in its nucleus. But we also talk about mass number, which is the sum of the protons and the neutrons combined. Because electrons have so little mass, we generally ignore them when calculating the weight of an atom. Usually, the number of protons and neutrons in an atom is equal, but nature allows for subtle variations. This brings us to the fascinating concept of isotopes. Imagine a family of siblings who all share the same last name, let's say carbon, and they all have the same 6 protons. However, one sibling might be a little heavier than the others because they have an extra neutron. We call these different versions of an element isotopes. While they behave almost identically in chemical reactions, their physical stability can differ. Some isotopes are stable, while others are radioactive, meaning their nucleus is a bit crowded and uncomfortable. Over time, these radioactive isotopes decay, giving off particles and energy as they seek a more stable state. Consider the history of how we learned to use these ticking clocks. In the mid-20th century, scientists like Willard Libby realized that carbon-14, a radioactive isotope, could be used to determine the age of ancient organic materials. Because carbon-14 decays at a very steady, predictable rate, we can measure how much of it remains in a fossil to tell the story of when that organism lived. It is as if every living thing carries a tiny atomic hourglass within them, counting down the millennia. Furthermore, we must give credit to the early thinkers who amassed these invisible truths. In the late 1700s, Antoine Lavoisier, often called the father of modern chemistry, proved that air was not a single substance, but a mixture of distinct gases, including oxygen and nitrogen. He was the one who insisted on careful measurement, transforming chemistry from a world of alchemy and guesswork into a rigorous science. A few decades later, John Dalton proposed the first modern atomic theory, suggesting that each element consists of unique atoms that combine in simple, whole number ratios. These discoveries were the first maps of the microscopic world we are navigating tonight. If your mind is still following along, Try visualizing the sheer scale of this world. If an atom were expanded to the size of a massive football stadium, the nucleus would be the size of a small marble sitting in the very center of the field. The electrons would be like tiny gnats buzzing around the very highest seats in the stands. Everything in between, the entire volume of the stadium, is just empty space. We only perceive the world as solid because the electrical charges of the electrons repel each other so strongly that they won't let us pass through. We are held up by the strength of invisible fields. Don't worry about memorizing these scales. Just let the wonder of it wash over you. The universe is built on a foundation of order and incredible lightness. You might wonder why we spend so much time discussing the weight of atoms or the behavior of isotopes. But the truth is, the balance of these elements is what determines your health and your very survival. Now, beyond the big four elements that make up the bulk of your body, there is another group called trace elements. These are elements that you need in only minute amounts, sometimes less than 0.01% of your body mass. But without them, the entire biological system can falter. Consider the element iodine. You only need a tiny speck of it, yet it is absolutely essential for your thyroid gland to produce the hormones that regulate your metabolism, growth, and development. In parts of the world where iodine is missing from the soil and diet, people can develop a goiter, an enlargement of the thyroid, as it struggles to do its job without its necessary ingredient. This is why many countries add a small amount of iodine to table salt, a simple, elegant chemical solution to a global health challenge. Similarly, we can look at the element iron. It makes up only a fraction of your weight, yet it sits at the very heart of the hemoglobin molecule in your blood. It is the specific magnetism of the iron atom that allows your blood to grab onto oxygen in your lungs and carry it to your brain and every other cell. If you have too little iron, you feel tired and weak, a condition we call anemia. You are essentially a machine that runs on the precise coordination of these minerals. So you see, this isn't just theory found in a dusty textbook. It is the principle that explains why we eat what we eat and how our bodies interact with the earth. We are literally composed of the Earth's crust and the atmosphere, organized into the most complex structures imaginable. Every time you take a breath of oxygen or season your food with salt, you are participating in the ancient, ongoing chemistry of the universe. You are the point where the chemistry of the stars meets the biology of the planet. This is a good place to rest your thoughts for a moment before we continue our journey. Think about how sturdy you feel as you lie there, and then remember the stadium of the atom. Think about how simple a breath of oxygen is, and then remember how it becomes the very water that fills your cells. Take a moment to let those ideas find their place in your mind, like stones settling at the bottom of a clear, still pond. We have now identified our cast of characters, the elements, and the individual units of their existence, the atoms. We have seen how they hold their identities through their protons, and how they offer variety through their isotopes. We have also seen the magic of emergence, in where simple elements combine to form compounds with entirely new lives. But in a world made only of lone atoms and static compounds, life could not exist. Life requires movement, interaction, and stability. The real story begins when these atoms start to reach out to one another. They don't just bump into each other like marbles in a jar. They connect, they share, and they trade. They form bonds that are strong enough to build mountains, yet flexible enough to be broken in an instant when your body needs energy. Having now grasped the structure of the individual atom, we are perfectly prepared to appreciate its social life, how it interacts with its neighbors to build the world. So when we return, we will take the next step together and explore the invisible handshakes of the molecular world. Let's transition our thoughts toward our next study, which is the study of atomic structure and chemical bonds. Sleep deeply and know that the foundations are secure. Lesson 2: Atomic Structure and Chemical Bonds. In our first session, we introduced the alphabet of existence, the elements, and the atoms that define them. We visualize the atom as a quiet, vast cathedral, mostly empty space, with a dense nucleus at its heart. But if atoms simply existed as isolated spheres, the universe would be a very lonely and static place. There would be no mountains, no oceans, and certainly no biological life. The beauty of our world arises from the fact that atoms are social. They have a deep, underlying drive to connect with one another. Tonight, we explore the invisible glue of the biological world. We are moving from the study of the individual to the study of the relationship. We will look at why some atoms are drawn together like magnets, and while others share their very essence to create the sturdy backbones of your DNA. This is the story of the chemical bond, the handshake of the molecular world. As we delve into these attractions, remember that there is no need to strain. Just let the logic of these connections settle into your mind as you relax. To understand why atoms bond, we must look away from the nucleus for a moment and focus our attention on the very edges of the atom, the electron cloud. While the protons in the center determine who an atom is, it is the electrons on the outside that determine how an atom behaves. Specifically, we are interested in the electrons that reside in the outermost shell, or energy level. These are known as valence electrons. You might imagine the energy levels around a nucleus as concentric circles, much like the layers of an onion. The layer closest to the nucleus is small and can only hold 2 electrons. The layers further out are larger, and for the elements most important to life, they generally seek to hold 8 electrons. This is a fundamental rule of thumb, in chemistry often called the octet rule. There is a profound sense of completeness, or satisfaction, an atom feels when its outermost shell is full. Most atoms in their natural, isolated state are not full. They are restless, looking for a way to reach that stable state of 8. This restlessness is the driving force behind all chemical reactions. Atoms will gain, lose, or share electrons with their neighbors until they achieve a full outer shell. This process of seeking stability creates the chemical bond. There are 2 primary ways this happens in the biological world, and they represent 2 very different types of relationships. The first is the ionic bond. Imagine an atom that has only 1 electron in its outer shell, perhaps a sodium atom. It is much easier for that atom to simply give away that 1 lonely electron than it is to go find 7 more. On the other side of the room, imagine a chlorine atom that has 7 electrons in its outer shell. It is desperately searching for just 1 more to reach the magic number of 8. When these 2 meet, a transfer occurs. Sodium gives its electron to chlorine. But remember, electrons carry a negative charge. When sodium loses a negative electron, it becomes positively charged. When chlorine gains that negative electron, it becomes negatively charged. These newly charged atoms are called ions because opposites attract. The positive sodium and the negative chlorine snap together in an intense electrical embrace. This is the ionic bond. It is not a physical link, but an irresistible attraction between two charged particles. The second, and perhaps more vital bond for the structure of life, is the covalent bond. This is not a story of giving and taking, but a story of sharing. Imagine two atoms that both need an electron to be complete. Instead of one stealing from the other, they move close together until their electron clouds overlap. They decide to share a pair of electrons, which then zip around both nuclei, acting as a physical tether that holds the atoms together. This shared pair of electrons creates a molecule. Unlike the ionic bond, which can be easily disrupted by something like water, the covalent bond is incredibly strong and stable, and it is the structural steel of the biological world. When you think of the long, elegant chains of your proteins or the spiraling ladder of your DNA, you are looking at the strength of covalent bonds at work. They allow life to build complex, permanent structures that can withstand the chaos of the environment. As we look deeper into the covalent bond, we find that not all sharing is created equal. This brings us to a concept that is essential for understanding why you are mostly made of water: electronegativity. Think of electronegativity as an atom's appetite for electrons. Some atoms, like oxygen, are very greedy. They pull on shared electrons with a much stronger force than atoms like hydrogen. When two atoms of the same element share electrons, like two oxygen atoms joining together, the pull is equal. The electrons spend an equal amount of time around each nucleus. We call this a nonpolar covalent bond. It is a perfect symmetrical partnership. However, when oxygen bonds with hydrogen to form water, the oxygen atom pulls the electrons much closer to itself. This unequal sharing creates a polar covalent bond. Because the negative electrons spend more time near the oxygen, that end of the molecule develops a slight negative charge. Conversely, because the electrons are pulled away from the hydrogen atoms, those ends develop a slight positive charge. The molecule itself is still neutral overall, but it has poles, a positive end and a negative end, much like a tiny battery or a magnet. This polarity is the secret reason water is the cradle of life, a topic we will explore in our next lesson. While ionic and covalent bonds are the strong bonds that build molecules, life also relies on several types of weak bonds. You might wonder why something weak would be important, but in biology, flexibility is just as important as strength. The most famous of these is the hydrogen bond. This occurs when the slightly positive hydrogen atom of one polar molecule is attracted to the slightly negative atom of another polar molecule. Think of a hydrogen bond not as a permanent weld, but as a Velcro connection. It's strong enough to hold things together, but weak enough to be pulled apart when necessary. This is how the two strands of your DNA stay together. They are held by millions of these tiny Velcro-like hydrogen bonds. When your cell needs to read your genetic code, it can easily unzip the DNA because the bonds are weak. And if they were held by strong covalent bonds, the cell would have to use a massive amount of energy just to open the book of life. Historically, our understanding of these bonds has evolved tremendously. In the early 20th century, the chemist Gilbert N. Lewis introduced the idea of the Lewis dot structure, which helped us visualize these valence electrons as dots around an element symbol. This simple tool allowed scientists to predict how atoms would bond long before we had the technology to actually see the electron clouds. It was a triumph of logic over the invisible. Take a moment to let that sink in. Your very existence depends on the greed of oxygen atoms and the generosity of hydrogen. The complexity of your body is managed by a delicate balance of permanent welds and temporary Velcro. If your mind is still following, you try to visualize those trillions of water molecules in your body, all constantly reaching out to one another with these brief, flickering hydrogen bonds, creating a fluid, shimmering web of life. The principles of chemical bonding are not just confined to textbooks. They are the reason the world feels and acts the way it does. You can see the difference between ionic and covalent bonds in your own kitchen. Consider a grain of table salt, which is held together by ionic bonds. When you drop it into water, the polar water molecules easily pull the ions apart, and the salt disappears or dissolves. The bond is strong when dry, but vulnerable in the presence of water. Now, consider a piece of plastic or the fiber in a celery stick. These are held together by covalent bonds. When you put them in water, they don't dissolve. The atoms are locked in a shared embrace that water cannot easily break. This is why our bodies use covalent bonds to build our cell walls and our bones. We need structures that won't simply melt away when we go for a swim. Bonding also explains the miracle of medications. Many drugs work because they are shaped exactly like the molecules your body naturally uses. A drug might have the perfect arrangement of polar and nonpolar regions to bond temporarily to a protein in your brain, mimicking a natural signal or blocking a harmful one. Pharmacists and chemists spend years studying the electronegativity and bond angles of atoms to design molecules that fit into the locks of your biological system. Even the air you breathe is a lesson in bonding. You don't breathe single oxygen atoms. They are too reactive and unhappy to be alone. And you breathe O2, two oxygen atoms locked in a double covalent bond, sharing 4 electrons to reach that state of perfect stability. It is this stable bonded form that your lungs are designed to capture. So as you rest tonight, consider the incredible coordination happening within you. Every time a muscle twitches or a thought forms, it is the result of millions of chemical bonds forming and breaking in a perfectly timed dance. You are a masterpiece of chemical architecture, held together by the simple, elegant need for atoms to find completeness in one another. We have now explored the glue that holds the universe together. We've seen how atoms move from isolation to community through the transfer and sharing of electrons. We've learned that the strength of these bonds creates the structures of life, now while their polarity creates the nuances of character. One specific molecule we mentioned, the water molecule, is perhaps the most extraordinary result of these bonding rules. Because of its polar covalent bonds and the resulting hydrogen bonds, water behaves unlike any other substance on Earth. It is the medium in which all the chemistry we've discussed actually takes place. Let's hold that thought here. A complete piece of the puzzle. We have our atoms, and we know how they stick together. Now, We must look at the liquid world they inhabit. Our next lesson will hand us the adjacent piece, which is the study of the properties of water. Allow yourself to drift now, carried by the quiet logic of the molecular world. There is no more to do tonight but rest. When we return, we will look at the blue heart of life. Lesson 3. The properties of water. As we continue our journey into the chemical foundations of life, we find ourselves standing before a substance so common that we often take it for granted, yet so extraordinary that without it, our planet would be as barren as a desert moon. We have already explored the individual atoms and the bonds that tie them together. We've learned that when oxygen, the greedy electron seeker, meets hydrogen, they form a polar covalent bond. This simple act of unequal sharing gives birth to a molecule that is, quite literally, the cradle of life. Tonight, we step into the blue heart of biology. We are going to look at water not just as a liquid we drink, but as a biological miracle with a unique set of talents. These talents are not accidental. They are the direct result of the chemical handshakes we discussed in our last session. Because of its shape and its electrical charges, water behaves unlike any other substance in the universe. It is the solvent for our cells, the regulator of our climate, and the medium in which every thought you have ever had was born. As we explore these properties together, let the concepts flow over you like a gentle stream. There is no need to grasp at every detail. Simply listen to the logic of how one simple molecule makes the vast complexity of life possible. To understand water, we must first visualize its shape. Imagine a head with two large ears, something like a famous cartoon mouse. The head is the large oxygen atom, and the ears are the two smaller hydrogen atoms. Because of the way the electrons are distributed, the ears are tucked toward one side, giving the molecule a distinct V-shape. As we learned, oxygen is highly electronegative. It pulls the shared electrons toward itself. This makes the oxygen end of the molecule slightly negative, and the hydrogen ends slightly positive. This polarity is the key to everything. Because water molecules have a positive and a negative end, they act like tiny magnets. When water molecules are near each other, the positive hydrogen of one molecule is attracted to the negative oxygen of another. This fleeting, flickering attraction is the hydrogen bond. While a single hydrogen bond is weak, much like a single thread of silk, millions of them acting together create a powerful and flexible web. This web gives water its 4 emergent properties that facilitate life. The first of these properties is cohesion. Because water molecules are constantly reaching out to one another through hydrogen bonds, the liquid stays together. You see this when you look at a dewdrop on a leaf. The water doesn't spread out into a thin film, but pulls itself into a bead. Closely related is adhesion, where water molecules use their sticky nature to cling to other substances, like the cell walls of a plant. Imagine a giant redwood tree, hundreds of feet tall. It has no pump, no heart to push water from the soil to the very top. Instead, it relies on a tug-of-war chain of water. Through adhesion, water clings to the narrow tubes inside the tree, and through cohesion, the water molecules hold onto each other like children playing crack the whip. As water evaporates from the leaves, it pulls the entire column of water upward, one molecule at a time, against the force of gravity. It is a silent hydraulic miracle happening in every garden and forest. The second property is water's remarkable ability to moderate temperature. Water is a thermal sponge. It can absorb or release a large amount of heat with only a slight change in its own temperature. This is because, in order to make water hotter, you first have to break those flickering hydrogen bonds before the molecules can start moving faster. Think of the ocean in the summer. The air might be scorching, but the water remains cool and steady. This high specific heat allows the oceans to store heat during the day and release it slowly at night, keeping the Earth's climate within a range that life can tolerate. The third property is perhaps the most counterintuitive: expansion upon freezing. In almost every other substance, the solid form is denser than the liquid form. If you freeze a block of lead, it sinks. But water is different. As water cools, the molecules move more slowly, and those hydrogen bonds begin to form a rigid, crystalline lattice. In this frozen state, the molecules are actually pushed further apart than they were in the liquid state. This makes ice less dense than water, which is why it floats. Finally, we must consider water as the universal solvent. Because it is polar, water is incredibly good at dissolving other polar substances and ionic compounds, like salts and sugars. The water molecules surround each individual ion or molecule, shielding them and pulling them into the solution. This creates a hydration shell. Within your body, this means that your blood is not just a fluid, but a complex chemical soup where nutrients, hormones, and gases are all dissolved and ready to be transported where they are needed. If your mind is still following the flow of these ideas, let's look a little deeper at the history and the why behind these properties. For a long time, early scientists struggled to explain why water was so different from other similar molecules, such as methane, CH4. Methane is about the same size as water, yet it is a gas at room temperature and freezes at an incredibly low temperature. The difference, we eventually realized, is that methane is nonpolar. It doesn't have those sticky hydrogen bonds. Without hydrogen bonding, the Earth's water would all be a gas, and our oceans would simply drift away into space. Consider again the floating of ice. This isn't just a curiosity for ice cubes in a glass. It is a fundamental requirement for the survival of the biosphere. In the late 19th century, naturalists began to observe that if ice sank, ponds, lakes, and even the great oceans would freeze from the bottom up. During the winter, the sun would only be able to melt a thin layer on the top, while the depths remained a solid, frozen tomb. Life as we know it would be crushed and frozen out of existence, but because ice floats, it forms an insulating blanket on the surface. This blanket traps the heat below, allowing fish, plants, and microorganisms to survive in the liquid water beneath the ice, even in the harshest winters. We should also reflect on the concept of evaporative cooling. This is the process that happens when the fastest moving, and therefore hottest, molecules of water escape from a surface as gas. As they leave, they take their heat energy with them, leaving the remaining surface much cooler. This is why you sweat. Your body isn't just getting wet, it is using the high heat of vaporization of water to pull excess heat away from your skin. It is the same principle that keeps a tropical forest from overheating. The evaporation of water from the leaves of trees acts like a massive planetary air conditioning system. Don't worry about the math of specific heat or joules of energy. Just let the image of water as a protective, stabilizing force find a place in your thoughts. It is the buffer that keeps the world from swinging into chaotic extremes of temperature. It is the medium that holds us steady. The properties of water aren't just abstract biological rules. They are the reason our world functions the way it does. You see the effect of water's high specific heat every time you look at a weather map. Coastal cities like San Francisco or London have much milder climates than the cities located deep in the middle of a continent, like Chicago or Moscow. The vast body of water nearby acts as a giant thermostat, absorbing the summer heat and giving it back during the winter. We are, in a very real sense, sheltered by the thermal inertia of the sea. You can also see the power of surface tension, a result of cohesion, when you watch a water strider walk on the surface of a pond. The hydrogen bonds are so strong that they create an invisible skin on the water that can support the weight of a small insect. It is a reminder that the weak bonds of chemistry can create very real physical boundaries in our world. In the world of medicine and health, the universal solvent property is what allows for the miracle of hydration and the delivery of medicine. When you take a pill, your body relies on the water in your stomach and blood to dissolve those molecules and carry them through the intricate plumbing of your veins and arteries. Even the way our cells communicate depends on water. The space between your brain cells is filled with a watery fluid that allows chemical signals to drift from one neuron to the next. Without that watery bridge, your thoughts would never reach their destination. Furthermore, we must mention the pH scale, which is a measure of the acidity or alkalinity of a solution. This is entirely based on the behavior of water molecules. Occasionally, a water molecule, H2O, will actually fall apart and dissociate into a hydrogen ion, H+, and a hydroxide ion, OH-. Though this happens rarely, the balance of these ions is incredibly important. If a substance adds more H+ to the water, it becomes an acid. If it adds OH-, it becomes a base. Your blood must maintain a very narrow, slightly basic pH, around 7.4, to stay healthy. Your body uses buffers to keep this balance, ensuring that the chemistry of your life doesn't become too sour or too bitter. So you see, water is not just a passive background. It is an active participant in every breath you take. It is the tugging force in the trees, the cooling mist on your skin, and the protective blanket on a frozen lake. We have now spent this hour together exploring the fluid environment of life. We've seen how the stickiness of water molecules creates the conditions for life to survive extremes, to transport nutrients, and to build the magnificent structures of the natural world. Water is the stage upon which the drama of life is performed. But a stage needs a framework. If water is the medium, we still need the sturdy, complex scaffolding that gives a cell its shape and its specialized functions. For that, we turn away from the fluid and toward the solid. We turn toward an element that is so versatile, it can build millions of different shapes, from the fuel in your cells to the information in your genes. Let's hold that thought here, a complete piece of the puzzle. Our next lesson will hand us the adjacent piece, which is the study of the element that serves as the backbone of every organic molecule. When we return, we will begin our study of carbon and the molecular diversity of life. Rest deeply now. Imagine the steady, rhythmic movement of the tides governed by the same simple laws of attraction we have discussed tonight. Your body is a vessel of this ancient, blue wisdom. Sleep well. Lesson 4. Carbon and the molecular diversity of life. In our journey thus far, we have explored the fundamental alphabet of the universe and the invisible glue of chemical bonds. We have marveled at the unique, life-sustaining properties of water, the fluid stage upon which the drama of existence unfolds. But a stage, no matter how perfect the medium, requires a lead actor. It requires a substance capable of building complexity, structure, and variety. To find this, we turn our attention away from the fluid and toward the solid framework of life. Tonight, we are going to explore the remarkable versatility of the element carbon. Carbon is the undisputed backbone of every organic molecule. While water provides the environment for life, carbon provides the scaffolding. It is the architect of the biological world, capable of forming millions of different shapes, from the simple fuel that powers your brain to the incredibly complex instructions found within your DNA. As we delve into the molecular diversity of life, I want you to imagine carbon not just as an element on a chart, but as a master builder with an infinite set of blueprints. There is no need to rush. Simply let the logic of this structural elegance wash over you as we move through our next chapter together. To understand why carbon is so central to life, we must look once again at its atomic structure. You may remember that atoms seek a state of completeness by filling their outermost electron shell. Carbon has an atomic number of 6, which means it has 6 protons and 6 electrons. 2 of those electrons sit in the inner shell, leaving 4 electrons in its outer, or valence, shell. Because this outer shell needs 8 electrons to be full, carbon has 4 vacancies. In the language of chemistry, we say carbon has a valence of 4. Imagine an atom with 4 arms reaching out in every direction, searching for partners to share electrons with. This tetravalence is carbon's greatest gift. Because it can form 4 covalent bonds simultaneously, it can build molecules of immense size and complexity. It can bond with other carbon atoms to create long, sturdy chains, or it can branch out like the limbs of a tree, or even fold back on itself to form stable rings. This ability to bond with itself and a variety of other elements, most notably hydrogen, oxygen, and nitrogen, is what makes the molecular diversity of life possible. When we speak of organic chemistry, when we are essentially speaking of the study of carbon compounds. The simplest of these are hydrocarbons, which consist only of carbon and hydrogen. You might picture a hydrocarbon as a simple skeleton. While hydrocarbons are rare in living organisms themselves, mostly found in things like petroleum or the fats that store our energy, they provide the underlying structure for more complex molecules. The energy stored in the bonds between carbon and hydrogen is the very same energy that fuels your cells and, in a much different context, powers a combustion engine. However, a skeleton alone does not make a functional body. To give these carbon backbones their specific personalities and biological functions, nature attaches various clusters of atoms known as functional groups. And these groups are the chemical accessories that determine how a molecule will behave in the water-filled environment of a cell. For instance, if you add a hydroxyl group, an oxygen bonded to a hydrogen, written as -OH, to a hydrocarbon, you turn it into an alcohol. This small change makes the molecule polar, allowing it to dissolve in water and participate in the chemistry of life. Other groups, like the carboxyl group found in organic acids, or the amino group found in the building blocks of proteins, add their own unique properties, making molecules acidic, basic, or reactive in specific ways. Through the endless combination of these carbon skeletons and functional groups, life produces a staggering array of molecules, each perfectly suited for a specific task. Take a moment to reflect on this. From one simple element with 4 empty spaces, nature builds the entire library of biological form and function. If your mind is still following along, let us step back and look at the history of how we came to understand this living chemistry. For centuries, scientists believed in a philosophy called vitalism. They thought that the organic molecules found in living things possessed a vital force that could not be explained by the physical laws of the universe. They believed that while you could study minerals and gases in a lab, you could never create a living molecule from dead matter. This belief began to crumble in 1828 when a German chemist named Friedrich Wöhler attempted to make an inorganic salt called ammonium cyanate. To his surprise, he instead produced urea, a compound naturally found in the urine of animals. He had created an organic molecule from inorganic starting materials. This was a profound moment in human history. It proved that the chemistry of life is not magical. It follows the same rules of bonding and energy as the rest of the physical world. A few decades later, Stanley Miller performed a famous experiment simulating the conditions of early Earth, showing that complex organic molecules could form spontaneously from simple gases and lightning. Another layer of carbon's complexity lies in the concept of isomers. Isomers are molecules that have the same chemical formula, the same number of carbon, hydrogen, and oxygen atoms, but different structures. It is like having a set of Lego bricks. You can use the same 20 bricks to build a tower, a bridge, or a small house. In biology, structure is everything. There are 3 main types of isomers. Structural isomers differ in how their atoms are covalently bonded together. Cis-trans isomers have the same bonds, but differ in their spatial arrangement, often around a double bond that prevents rotation. Finally, there are enantiomers, which are perhaps the most fascinating. Enantiomers are mirror images of each other, much like your left and right hands. You can see they are made of the same parts, but you cannot perfectly overlap your right hand onto your left. In the world of medicine, this distinction is a matter of life and death. One enantiomer of a drug might be a powerful medicine, while its mirror image might be completely inactive or even harmful. Your body's receptors are so specific that they can tell the difference between these left-handed and right-handed molecules. This reminds us that in the realm of carbon chemistry, the where and how of an atom's placement is just as important as the what. The versatility of carbon isn't just a topic for the laboratory. It is the reason you are able to interact with the world around you. Every time you eat a piece of fruit, you are consuming carbon chains built by a plant using nothing but sunlight, water, and the carbon dioxide from the air. The plant uses carbon to build cellulose, a tough polysaccharide that gives its stalks and leaves their strength. When you eat that fruit, your body breaks those carbon bonds apart to harvest the energy stored within them, using those same carbon atoms to build your own muscles, skin, or in hair. This cycle, the carbon cycle, is the great circulatory system of our planet. Carbon moves from the atmosphere into living things, and then back into the earth or the air when those living things breathe or decompose. We are part of a continuous, ancient recycling project. The carbon atom that is currently part of your DNA might have once been part of a dinosaur's tooth or a leaf on an ancient fern. Carbon is the ultimate traveler, moving through time and form, always maintaining its four-armed reach for connection. We also see the impact of carbon's diversity in the field of synthetic chemistry. Because we understand the rules of carbon bonding, we can create plastics, medicines, and fibers that do not exist in nature. However, this also presents a challenge. Many of our synthetic carbon compounds, like plastics, are held together by such strong covalent bonds that natural bacteria cannot break them down. This is the flip side of carbon's stability. The same strength that makes a molecule useful can also make it a persistent pollutant. Consider, too, the role of carbon in global climate. Carbon dioxide, CO2, is a simple molecule, one carbon atom double bonded to two oxygen atoms. Despite its simplicity, its ability to absorb and re-emit infrared radiation is what keeps our planet warm enough for liquid water to exist. However, as we burn fossil fuels, which are essentially ancient, buried sunshine, trapped in carbon bonds, we release that stored carbon back into the atmosphere, shifting the delicate balance that life has relied on for millennia. Understanding carbon is not just about understanding our bodies, it is about understanding our responsibility to the planet. We have now spent this time appreciating the architect. We've seen how carbon's four valence electrons allow it to serve as the structural foundation for an almost infinite variety of molecules. We've learned about the functional groups that give these skeletons their purpose, and the isomers that give them their specific identity. But a framework needs a purpose. If carbon provides the scaffolding, what are the actual buildings it creates? In the world of biology, we call these giant molecules macromolecules. These are the four great classes of organic compounds that make up every living cell. To understand how life actually works, we must now look at these structures one by one. Let's hold that thought here, a complete piece of the puzzle. We have our architect and our blueprints. Now, we must look at the first great category of construction, the molecules of energy and structure. Our next lesson will lead us into the world of sugars and fibers as we study the structure and function of carbohydrates. Rest your mind now. Visualize those tiny carbon atoms reaching out their four arms, holding the world together in a silent, sturdy embrace. There is no more to do tonight but find your own state of stability. When you are ready, we will continue our journey through the molecular landscape. Lesson 5. Structure and function of carbohydrates. In our previous session, we spent time with the architect of life, carbon. We saw how its four-armed reach creates the scaffolding for everything we are. But a scaffold is not yet a home, and a blueprint is not yet a life. Tonight, we move from the framework to the first great class of biological buildings, the carbohydrates. When you hear the word carbohydrate, you might immediately think of a loaf of warm bread, a bowl of pasta, or perhaps the sweetness of a piece of fruit. While those are certainly part of the story, in the world of biology, carbohydrates are far more than just fuel. They are the sturdy walls of a towering oak tree, the protective armor of a beetle, and the tiny identity tags on the surface of your cells that tell your immune system who you are. Carbohydrates represent a beautiful marriage of energy and structure. They are the primary way that life captures the energy of the sun and stores it for a rainy day. As we explore these elegant molecules together, I want you to imagine them as a series of connected links in a chain, sometimes short and simple, sometimes vast and complex. There is no rush to memorize every bond. Simply let the logic of how sugars build the world wash over you as you relax into your rest. To understand carbohydrates, we begin with their name, which literally means carbon water. Most carbohydrates follow a simple mathematical rhythm. For every carbon atom, there are 2 hydrogen atoms and 1 oxygen atom. This CH2O ratio is the foundational pulse of the group. We categorize these molecules based on their size and complexity, now starting with the simplest units, which we call monosaccharides. A monosaccharide is a single link in the carbohydrate chain. The most famous of these is glucose, a molecule that is quite literally the cash currency of your body. If you think of your metabolism as an economy, glucose is the dollar bill, easy to spend, universally accepted, and essential for keeping the lights on. In a laboratory, we might draw glucose as a straight line of carbons, but in the watery world of your cells, it almost always folds into a stable, 6-sided ring. This ring structure is vital because it creates a solid, predictable shape that enzymes can recognize and process. When 2 of these single sugars find each other and decide to bond, they form a disaccharide. This happens through a process we call a dehydration reaction, where 2 molecules come together by donating the pieces to form a water molecule, which then drifts away. The bond that holds them together is called a glycosidic linkage. You encounter these every day. For example, when a glucose and a fructose come together, they form sucrose, which is the common table sugar you might stir into your tea. It is a simple, elegant pairing of 2 individual units. However, the true power of carbohydrates is revealed when we move from pairs to thousands. These are the polysaccharides, the giant macromolecules of the sugar world. You can imagine these as long, winding necklaces, made of hundreds or even thousands of glucose beads. Depending on how those beads are linked together, and whether the chain branches out like a tree, on the polysaccharide will serve one of 2 primary roles: storage or structure. Storage polysaccharides are the savings accounts of the biological world. Plants build starch, a long chain of glucose molecules coiled into a spiral. When the sun isn't shining, the plant can reach into this account, snip off a few glucose beads, and use them for energy. In humans, we do something similar with a molecule called glycogen. We store it in our liver and muscle cells. When you go for a long walk, or haven't eaten in a few hours, your body begins to break down glycogen, releasing a steady stream of glucose into your blood to keep your brain and muscles powered. It is a perfect system of biological foresight. If your mind is still drifting along with the logic of these chains, let us look at the second role of polysaccharides, structure. And this is where the subtle geometry of chemistry becomes fascinating. You might wonder why we can eat a potato which is full of starch and get energy from it. But if we try to eat a piece of wood or a blade of grass, which is full of cellulose, we get almost no energy at all. After all, both starch and cellulose are made of the exact same ingredient, glucose. The difference lies in the orientation of the glucose molecule. Imagine a line of people holding hands. In starch, everyone is facing the same way, creating a flexible coiled chain that is easy for our digestive enzymes to unzip. In cellulose, however, every other glucose molecule is flipped upside down relative to its neighbor. This simple flip allows the chains to lie perfectly flat and straight. These straight chains then huddle together and form hydrogen bonds with one another, creating incredibly tough, stiff fibers called microfibrils. This is the material that makes up the cell walls of plants. It is the reason a tree can stand hundreds of feet tall without a skeleton. It is one of the most abundant organic compounds on Earth. When we eat plants, we call this cellulose fiber. Because our enzymes are designed to only recognize the everyone facing the same way bond of starch, they simply cannot break the flipped bond of cellulose. The molecule passes through our system untouched. It is a humble reminder that in biology, the way things are put together is just as important as what they are made of. Historically, our understanding of these sugars was transformed by the work of Emil Fischer in the late 19th century. He was the first to map the three-dimensional shapes of sugars, winning the Nobel Prize for proving how even a tiny change in the position of a single hydroxyl group could change one sugar into another. He showed us that the molecular world is a world of precise spatial puzzles. Beyond starch and cellulose, there is another remarkable structural carbohydrate called chitin. This is what makes up the crunchy exoskeleton of insects, spiders, and crustaceans. It is also found in the cell walls of fungi. Chitin is similar to cellulose, but it has a nitrogen-containing attachment on each glucose unit that makes it even tougher. It is a testament to the versatility of the carbohydrate blueprint. With just a few small tweaks, a molecule used for energy can become a suit of armor. The story of carbohydrates is not just one of energy and armor. It is also a story of communication. If you could look at the surface of one of your own cells through a powerful microscope, you wouldn't see a smooth surface. Instead, you would see a forest of tiny, branching sugar chains attached to proteins and lipids. We call these glycoproteins and glycolipids. These sugar chains act like the ID tags of the body. They are how your cells recognize one another. For example, the reason you have a specific blood type, AB, AB, or O, is entirely due to the specific arrangement of carbohydrate molecules on the surface of your red blood cells. If you receive the wrong blood type during a transfusion, your immune system looks at those foreign sugar tags, realizes they don't belong to you, and attacks them. Much of the complexity of our immune system and how our cells stick together to form tissues is governed by this sugar code. We also see the importance of carbohydrates in our modern understanding of health. We often hear about simple versus complex carbohydrates. Simple carbs, like the sucrose in candy or the lactose in milk, are made of 1 or 2 units. They are broken down almost instantly, causing a sharp spike in blood sugar. Complex carbs, like the starches in whole grains and vegetables, take much longer to dismantle. They provide a slow, steady release of energy, which is much easier for the body to manage. Even the feeling of hitting the wall that marathon runners describe is a carbohydrate story. It happens when the body has completely used up its savings account of glycogen in the liver and muscles. Without those glucose chains to snip apart, the body has to switch to burning fats, which is a much slower process, causing the athlete to feel a sudden and profound fatigue. So you see, carbohydrates are the bridge between the sun and your own heartbeat. They are the energy of the past stored in starch, the strength of the present in your cell membranes and the plants around you, and the instructions for the future as they help your cells organize and grow. You are, in many ways, a living expression of the energy stored in these elegant carbon rings. We have now spent this hour exploring the world of sugars, from the single energetic ring of glucose to the massive structural cables of cellulose. We've seen how life uses carbohydrates to spend energy, to save energy, and to build the walls of the natural world. And they are the cash and the bricks of the biological economy. But life requires more than just energy and structure. It also needs a way to store energy for the long term, not just for a few hours, but for weeks or months. It needs a way to create waterproof barriers and to send chemical signals over long distances. For that, we turn to a group of molecules that famously refuses to mix with the watery world we've discussed so far. Let's hold that thought here. A complete piece of the puzzle. We have understood the sugars. Now, we must look at the outsiders of the molecular world. Our next lesson will take us into the water-fearing, energy-dense realm of fats and oils as we study the structure and function of lipids. Rest deeply now. Imagine the steady, patient growth of a tree, adding layer upon layer of cellulose as it reaches for the light. Your own body is quietly managing its glycogen stores as you sleep, ensuring you have the energy to wake and begin a new day. There is no more to do tonight. Lesson 6. Structure and function of lipids. As we continue our journey through the molecular landscape of life, we have so far moved through the fluid, sticky world of water and the versatile, ring-based world of carbohydrates. In our last session, we saw how sugars act as the immediate currency and the structural fiber of the living world. But tonight, we turn our attention to a group of molecules that seems to play by an entirely different set of rules. These are the lipids. While almost every other molecule we have discussed, from the ions of salt to the rings of glucose, interacts easily and even happily with water, lipids are the great outsiders. The defining characteristic of a lipid is its hydrophobicity, its fundamental aversion to water. You have seen this yourself every time you look at a bottle of vinaigrette and see the oil floating in distinct, shimmering beads above the vinegar. This refusal to mix is not just a culinary curiosity, it is one of the most important principles in all of biology. It is what allows a cell to have a boundary, what allows your nerves to send signals without leaking, and what allows your body to store energy for the long term in a compact, efficient way. Tonight, we explore the water-fearing world of fats, oils, waxes, and steroids. As we delve into the structure of these molecules, try to visualize them as the insulators and gatekeepers of your biology. Let the logic of their resistance to water settle into your mind as you find your own place of quiet and rest. To understand lipids, we must first address a unique distinction. Unlike carbohydrates, proteins, or nucleic acids, lipids are not true polymers. They are not built from long, repeating chains of identical building blocks like beads on a string. Instead, they are a diverse group of molecules, grouped together because they share one physical trait. They are poorly soluble in water. This is because they consist mostly of hydrocarbon regions, long chains of carbon and hydrogen that share their electrons equally, making them nonpolar. Since water is polar, it has no interest in interacting with these neutral chains. The first and most familiar group of lipids is the fats, also known as triglycerides. A fat molecule is constructed from two kinds of smaller molecules, a single alcohol called glycerol, and three long fatty acid chains. These fatty acids are essentially long tails of carbon atoms, usually 16 to 18 in length, with hydrogens crowding around them. When the three tails attach to the glycerol head, they form a shape that looks a bit like the capital letter E. The primary function of fats is energy storage. While a carbohydrate is great for a quick burst of energy, it is heavy because it carries a lot of water weight with it. Fats, however, are anhydrous. They pack together without water. This allows them to store more than twice as much energy in a single gram as a carbohydrate. If we stored all our energy as carbohydrates instead of fats, we would be far too heavy to move effectively. In this sense, fat is the body's high-density battery, a long-term energy reserve that is light, efficient, and vital for survival during times when food is scarce. The second, and arguably most important group for the existence of life, is the phospholipids. These molecules are cousins to the fats, but with a critical modification. Instead of three fatty acid tails, they have only two. The third spot on the glycerol head is taken up by a phosphate group, which carries a negative electrical charge. This creates a fascinating and conflicted molecule. The phosphate head loves water, it is hydrophilic, while the two fatty acid tails still hate water. They are hydrophobic. When you place phospholipids into a watery environment, they perform a beautiful act of self-organization. They don't need instructions from the cell. They simply follow the laws of chemistry. To protect their tails from the water while keeping their heads in contact with it, they form a double layer, or a bilayer. All the tails point inward, huddling together in a dry, oily interior, while the heads point outward toward the water. This phospholipid bilayer is the fundamental structure of every cell membrane on Earth. It is the skin of the cell, the boundary that separates the inside from the outside. Without this specific lipid architecture, the complex chem, if your mind is still following these oily trails, let us look closer at the texture of these fats. You have likely heard the terms saturated and unsaturated in discussions about nutrition. These terms refer to the structure of the hydrocarbon tails. If every carbon in the chain is connected to its neighbor by a single bond, the chain is saturated with as many hydrogen atoms as possible. These chains are straight and flexible. Because they are straight, they can pack together very tightly, like a stack of lumber. This is why saturated fats, like butter or lard, are solid at room temperature. However, if there is a double bond between two carbons in the chain, it creates a kink, or a bend in the tail. This chain is now unsaturated because it has fewer hydrogens than it could potentially hold. These kinks prevent the molecules from packing tightly together. Imagine trying to stack a pile of bent sticks. They will always have space between them. Because they cannot pack together, these fats remain liquid at room temperature. These are the oils, like olive oil or vegetable oil, that we find primarily in plants and fish. Historically, our understanding of these structures has influenced everything from medicine to the food industry. In the mid-20th century, a scientist developed a process called hydrogenation, where they took unsaturated vegetable oils and forced hydrogen into them to break the double bonds and straighten the chains. This turned liquid oils into solid fats, creating things like margarine and shortening. However, this process often created trans fats, which have a specific type of bond that the human body struggles to process, leading to significant health challenges. It is a reminder that when we tinker with the delicate geometry of a lipid tail, we change how it interacts with the machinery of our cells. Beyond fats and phospholipids, there is a third, very different category of lipids, the steroids. At first glance, a steroid doesn't look like a fat at all. Instead of long tails, it is made of 4 fused carbon rings. However, because it is composed almost entirely of carbon and hydrogen, it is hydrophobic, which earns it a place in the lipid family. The most famous steroid is cholesterol. While it often gets a bad reputation in popular culture, cholesterol is essential for life. It tucks itself into the phospholipid bilayer of your cell membranes, acting as a temperature buffer. It prevents the membrane from becoming too fluid when it's hot, and keeps it from freezing solid when it's cold. Furthermore, cholesterol is the mother molecule from which your body builds chemical messengers like estrogen and testosterone. These hormones are lipids, which means they can easily pass through the oily membranes of your cells to deliver their messages directly to the heart of the cell. This is why hormonal signals can be so powerful and long-lasting. They have an all-access pass to the cellular interior because of their lipid nature. The unique properties of lipids are visible in almost every corner of the natural world. Consider the feathers of a duck. Have you ever wondered why a duck can spend all day in a cold pond and stay perfectly dry and warm? It is because the duck has a special gland that produces a waxy lipid. The duck spends hours preening, spreading this wax over its feathers. Because the wax is hydrophobic, the water cannot penetrate the feathers. It simply beads up and rolls off. The duck is wearing a permanent molecular raincoat. In our own lives, we see the power of lipids in the way we clean our world. Since like dissolves like, water alone cannot remove the oily grease from a frying pan or the natural oils from your skin. To bridge the gap, we use soap. A soap molecule is essentially a tamed lipid. It has a long, hydrophobic tail that buries itself into the grease, and a hydrophilic head that stays in the water. When you rinse, the soap molecules form tiny spheres called micelles, trapping the oil in the center and allowing it to be washed away by the water. You are using chemistry to force a truce between two elements that naturally refuse to meet. Lipids also play a quiet, vital role in the function of your brain. Every one of your nerve cells is wrapped in a thick, fatty sheath called myelin. This myelin is made of specialized lipid-rich cells. Much like the plastic insulation on an electrical wire, the myelin sheath prevents the electrical signal from leaking out as it travels down the nerve. This allows signals to hop along the nerve at incredible speeds. In diseases where this lipid insulation is damaged, such as multiple sclerosis, the signals slow down or fail to reach their destination. We think and move at the speed of lipids. Even the way we perceive flavor is tied to this group of molecules. Many of the most aromatic and flavorful compounds in spices and herbs are fat soluble. This is why a bit of butter or oil in a recipe helps to carry the flavor. The lipids dissolve those aromatic molecules and deliver them more effectively to your taste buds. It is yet another way that these outsider molecules facilitate our connection to the world around us. So you see, lipids are far more than just a source of calories or a topic for a diet book. They are the insulators that keep our nerves firing, the waterproofers that keep our cells intact, and in the long-term energy that keeps us moving through the world. They are the silent, oily guardians of our biological integrity. We have now explored the world of the outsiders, the hydrophobic molecules that provide our boundaries and our long-term reserves. We've seen how the E shape of a fat stores energy, how the conflict of a phospholipid builds the cell membrane, and how the rings of a steroid send messages through our systems. But while lipids provide the boundaries and the energy, they aren't the primary doers of the cell. If a cell were a factory, the lipids would be the walls and the fuel in the basement. We still need the workers, the machines, and the engineers who actually perform the tasks of life, breaking down food, building new structures, and defending against invaders. For that, we must turn to the most diverse and versatile class of molecules in the entire biological kingdom. Let's hold that thought here, a complete piece of the puzzle. We have understood the fats and the boundaries. Our next lesson will take us into the incredibly complex, folded world of the cellular workhorses. We will study the molecules that are defined by their shape and their action. When we return, we will begin our study of the structure and function of proteins. Rest deeply now. Imagine the calm, protective barrier of your own cell membranes, holding everything in place, keeping the inside safe from the outside. Your body is a vessel of quiet, oily stability. There is nothing more to do tonight but surrender to the rest your body has earned. Lesson 7. Structure and function of proteins. As we venture further into the intricate architecture of the cell, we have already witnessed the fluidity of water, the structural energy of carbohydrates, and the protective, insulating boundaries of lipids. We have seen the stage, the scaffolding, and the fuel. But now, we arrive at the most dynamic and versatile characters in our biological story, the proteins. If the cell were a grand, bustling city, the lipids would be the walls and the roads, and the carbohydrates would be the power plants. Proteins, however, would be everything else. They are the construction workers, the architects, the couriers, the security guards, and the machines in the factories. In fact, the word protein comes from the Greek word proteos, which means of first importance. This name is well earned. There is almost no process within a living organism that does not depend on these molecules. They are the workhorses of life, defined not just by what they are made of, but by the incredible specific shapes they take. Tonight, we will explore how a simple string of molecules can fold itself into a masterpiece of biological machinery. As we delve into the 4 levels of protein structure, I want you to imagine a long, flexible ribbon being folded into an intricate piece of origami. A shape so precise that it can recognize a single atom in a sea of millions. Let the logic of this folding process guide you into a deeper state of relaxation. To understand proteins, we must start with their building blocks, the amino acids. there are only 20 different amino acids that life uses to build its proteins. You might think of these 20 as a molecular alphabet. Just as you can use 26 letters to write a short poem or a massive technical manual, a cell can use these 20 amino acids to build an almost infinite variety of proteins. Each amino acid has a similar core structure: a central carbon atom bonded to an amino group, a carboxyl group, and a simple hydrogen atom. What makes each of the 20 unique is the fourth bond, known as the R group or the side chain. The R group is the personality of the amino acid. Some R groups are simple, like a single hydrogen atom. Others are complex rings. Some love water, while others hide from it. Some carry a positive or negative charge, while others are neutral. When a cell builds a protein, it links these amino acids together into a long chain called a polypeptide. The bond that connects them, formed through that familiar dehydration reaction we've seen before, is called a peptide bond. A protein is not just a random string, however. It is one or more polypeptide chains that have been twisted, folded, and coiled into a very specific three-dimensional shape. We describe this complex shape using 4 distinct levels of organization. The first level is the primary structure. This is simply the unique sequence of amino acids in the chain. Imagine a long string of different colored beads. The order of those beads is the primary structure. Even a change in just one bead out of hundreds can completely alter the protein's final shape and its ability to function. This sequence is not accidental. It is dictated precisely by the instructions in your DNA. As the chain grows, it begins to interact with itself, now leading to the secondary structure. This level of folding is caused by hydrogen bonds, forming between the backbones of the amino acids. These interactions usually create one of 2 patterns: a delicate coil called an alpha helix, or a folded accordion-like structure called a beta-pleated sheet. You might picture these as the initial curls or folds in our piece of origami. They provide the basic texture of the protein. The third level, the tertiary structure, is where the protein truly finds its 3D form. This level of folding is determined by the interactions between the R groups, those personalities we mentioned earlier. Hydrophobic R groups will huddle together in the center of the protein to stay away from the surrounding water, while hydrophilic groups will reach outward. Positive and negative groups will snap together like magnets, and special disulfide bridges can form a permanent weld between certain amino acids. The result is a complex glob-like or fiber-like shape that is unique to that specific protein. Finally, some proteins have a quaternary structure. This occurs when 2 or more polypeptide chains, each with its own tertiary structure, come together to form one functional unit. A perfect example is hemoglobin, the protein that carries oxygen in your blood. It is made of 4 separate polypeptide subunits that huddle together to create a single, powerful oxygen-carrying machine. Take a moment to reflect on this. From a simple sequence of 20 beads, life builds 3-dimensional tools of breathtaking precision. If your mind is still following the twists and turns of these folding chains, let us consider the vital relationship between a protein's shape and its environment. Because a protein is held together by many weak interactions, like hydrogen bonds and electrical attractions, its shape is delicate. If the temperature gets too high, or if the acidity of the environment changes, these weak bonds can break. When this happens, the protein unravels and loses its functional shape in a process called denaturation. You see denaturation every time you cook an egg. The clear white of a raw egg is full of a protein called albumin, which is folded into a neat soluble globe. When you apply heat, the kinetic energy shakes those delicate bonds apart. The protein unravels and then tangles up with its neighbors, forming a solid, white, opaque mass. Once a protein is denatured, it can almost never return to its original shape. And it is a one-way trip from a functioning machine to a tangled mess. This is why a high fever can be so dangerous for the human body. If the temperature rises too much, the very machines that keep us alive can begin to unravel. Historically, the mystery of how proteins fold was one of the greatest challenges in science. In the 1950s, Linus Pauling discovered the alpha helix and beta-pleated sheet, earning him the Nobel Prize. He realized that the simple hydrogen bond was the secret to the protein's stability. Later, Christian Anfinsen performed a famous experiment showing that if you gently denature a small protein and then return it to its original environment, it will often fold back up into its correct shape all by itself. This proved that the information for the 3D shape is contained entirely within the 1D sequence of amino acids. The beads know exactly how they are supposed to huddle together. In some cases, however, proteins need a little help to fold correctly. The cell uses specialized proteins called chaperonins. Imagine a tiny hollow cylinder with a lid. An unfolded polypeptide chain enters this safety cage. The lid closes, and the protein is given a protected, quiet space to fold correctly without being bumped by other molecules. When the folding is complete, the lid opens, and the new machine is released into the cell. It is a beautiful example of how life provides a supportive environment for even its smallest components to find their true form. We must also touch upon what happens when folding goes wrong. Many of the most challenging diseases we face, such as Alzheimer's and Parkinson's, are associated with misfolded proteins. These proteins fold into an incorrect sticky shape that the cell cannot break down. Over time, these misfolded proteins clump together into plaques that interfere with the brain's ability to function. It reminds us that in the world of proteins, a single wrong turn in the folding process can have profound consequences for the entire organism. The diversity of protein function is truly staggering. One of the most important roles they play is as enzymes. Most chemical reactions in the body would happen too slowly to support life if they were left to their own devices. Enzymes act as biological catalysts, grabbing onto specific molecules and either pulling them apart or pushing them together. They do this with incredible speed. Some enzymes can process thousands of molecules every single second. Without the enzyme lactase, for instance, you could not digest the sugar in milk. The enzyme provides a perfect pocket that fits the milk sugar molecule exactly, stressing its bonds until they snap. Proteins also provide the physical structure of your body. Collagen is the most abundant protein in the human kingdom. It forms long, triple-stranded fibers that act like molecular rope. This rope provides the strength in your skin, the toughness in your tendons, and the framework for your bones. While collagen provides the strength, other proteins like actin and myosin provide the movement. These are the proteins in your muscles that slide past one another, allowing you to walk breathe, and even for your heart to beat. You are a being made of protein, moved by protein, and maintained by protein. We also rely on proteins for protection and communication. Your antibodies are highly specialized proteins that circulate in your blood. Each antibody has a specific binding site, shaped exactly like a part of a virus or bacteria. When it finds an invader, it snaps onto it like a pair of handcuffs, marking it for destruction by the immune system. Meanwhile, protein hormones like insulin travel through your blood to tell your cells when to take up sugar from the environment. They act as the messengers that coordinate the vast, complex economy of your metabolism. Even the way we perceive the world is a protein story. In your eyes, a protein called rhodopsin sits in your retina. When a single photon of light hits this protein, it changes shape instantly. That tiny shift in shape triggers an electrical signal that your brain interprets as sight. And every sunset you see and every word you read is made possible by the rapid, mechanical shifting of a protein's structure. So you see, proteins are the ultimate expression of the molecular diversity of life. They are the bridge between the static instructions of our DNA and the dynamic, breathing reality of our lives. They are the artisans and the tools, the message and the messenger. Everything you are doing right now, resting, breathing, processing these words, is being performed by a tireless army of folded ribbons. We have now explored the world of the workers, the incredibly diverse proteins that perform the tasks of life. We've seen how 20 amino acids create the infinite variety of shapes that allow for movement, structure, defense, and digestion. We've learned that in the protein world, shape is everything, and that shape is a delicate, hard-won masterpiece. But who tells the amino acids what order to stand in? Who holds the master blueprints for these incredible machines? If the proteins are the workers and the tools, there must be an archive, a silent, stable library where the instructions for every protein in your body are kept safe and passed down from one generation to the next. For that, we turn to the final great class of macromolecules. Let's hold that thought here, a complete piece of the puzzle. We have understood the machinery. Our next lesson will take us into the heart of the cell's information system. We will study the molecules of inheritance and the code of life itself. When we return, we will begin our study of the structure and function of nucleic acids. Rest deeply now. Imagine the trillions of proteins within you quietly performing their tasks, folding, building, and protecting, all while you sleep. Your body is a masterpiece of active, living architecture. Lesson 8. Structure and function of nucleic acids. We have traveled through the bustling world of the cell, meeting the workers, the builders, and the guardians. We have seen the proteins performing their tireless labor and the lipids maintaining the boundaries. But a city without a library or a factory without a set of master blueprints would eventually fall into chaos. Every action we have discussed so far, the folding of a protein, the storage of a sugar, the pumping of an ion, requires a specific set of instructions. Tonight, we enter the most sacred and stable archive of the biological world, the nucleic acids. These molecules are the keepers of the code. They are the threads of continuity that connect you to your ancestors and, eventually, to the generations that will follow you. While the other macromolecules we've studied focus on the here and now of survival, Nucleic acids focus on the forever of inheritance. They are the medium through which the story of life is written, stored, and shared. As we explore the elegant, twisting architecture of DNA and the versatile messaging of RNA, I want you to imagine a vast library where every book is a recipe for a part of you. Let the steady rhythmic logic of this genetic archive guide you toward a place of deep intellectual peace. To understand the nucleic acids, we must look at their individual links, which we call nucleotides. Just as a protein is a chain of amino acids, a nucleic acid is a long chain of these nucleotides. Every nucleotide is a tiny 3-part assembly. First, there is a 5-carbon sugar, which acts as the central hub. Second, might attach to one side of the sugar is a phosphate group. Third, attached to the other side is a nitrogenous base. This base is the letter in the genetic alphabet. There are 2 main families of nucleic acids, distinguished primarily by the sugar in their hub. In DNA, or deoxyribonucleic acid, the sugar is deoxyribose. In RNA, or ribonucleic acid, the sugar is ribose. This might seem like a small detail, but that one extra oxygen atom in the ribose sugar makes RNA more flexible and less stable than DNA, perfect for a temporary messenger, but not for a permanent archive. The real magic, however, lies in the nitrogenous bases. There are 4 types of bases in DNA: adenine, guanine, cytosine, and thymine. In RNA, thymine is replaced by a similar base called uracil. These bases are divided into 2 groups: the large, double-ringed purines, adenine and guanine, and the smaller single-ringed pyrimidines, cytosine, thymine, and uracil. When these nucleotides join together, they form a sugar-phosphate backbone. Imagine a long, sturdy rail made of alternating sugar and phosphate groups, with the nitrogenous bases sticking out like the teeth of a comb. But DNA does not exist as a single rail. It is almost always found as a double helix. Picture a long ladder that has been twisted into a spiral. The 2 side rails are the sugar-phosphate backbones, and the rungs of the ladder are made of pairs of nitrogenous bases meeting in the middle. These bases are held together by the same gentle, Velcro-like hydrogen bonds we discussed in our lesson on water. Because of their specific shapes and electrical charges, these bases are very particular about their partners. Adenine always pairs with thymine, and guanine always pairs with cytosine. This is the law of base pairing. This specific pairing is the secret to life's ability to copy itself. Because one side of the ladder is a perfect mirror or complement of the other, the cell can unzip the ladder and use each half as a template to build a brand new partner. This is how a single cell, during the development of a human being, can copy its entire library of 3 billion letters and pass it on to a daughter cell with almost perfect accuracy. It is a system of data storage and replication that is more efficient and reliable than any hard drive ever built by human hands. If your mind is still drifting along these spiraling ladders, let us consider the historical journey that led us to this discovery. For a long time, scientists weren't sure what carried the instructions for life. Many thought it was proteins because they were so complex and diverse. It wasn't until the mid-20th century that a series of elegant experiments proved that the transforming principle was actually DNA. In 1953, the structure of the double helix was finally unmasked. James Watson and Francis Crick are the names most often associated with this breakthrough, but their work would have been impossible without the contributions of Rosalind Franklin. Franklin was an expert in X-ray crystallography, a technique where you shine X-rays through a crystal of a molecule to see the pattern they make. In her photograph 51 was the crucial piece of evidence that showed the unmistakable X pattern of a helix. Watson and Crick realized that for the chemistry to work, the two strands of the ladder had to run in opposite directions, what we call anti-parallel, like two lanes of traffic on a highway. While DNA is the permanent master blueprint kept safe in the nucleus of the cell, RNA is the versatile working copy. RNA is typically single-stranded, which allows it to fold into various shapes, much like a protein. There are several types of RNA, each with a specific job in the central dogma of biology. This is the process where information flows from DNA to RNA, and finally to protein. First, there is messenger RNA, mRNA. When the cell needs a specific protein, it makes a temporary RNA copy of that gene. This messenger leaves the protected nucleus and travels to the protein-building machinery. Then there is transfer, RNA, tRNA, which acts like a molecular translator. It carries the correct amino acid to the messenger, matching its own code to the code on the mRNA. Finally, there is ribosomal RNA, rRNA, which actually builds the ribosome, the workbench where the protein is assembled. Take a moment to let that idea find its place. Your DNA is a silent library of possibilities. It doesn't do anything on its own. It requires the active, moving assistance of RNA to turn those silent instructions into the breathing, acting proteins that make you you. It is a perfect hierarchy of information management. We should also reflect on the sheer scale of this information. If you were to take the DNA from a single human cell and stretch it out, it would be about 2 meters long. To fit that much material into a microscopic nucleus, the DNA is wrapped around special proteins called histones, like thread around a spool. These spools are then coiled and folded again and again into the structures we call chromosomes. You have 46 of these books in almost every cell of your body. If you were to read the sequence of your DNA aloud at a rate of 1 letter per second, it would take you nearly 100 years to finish. Yet your cells can read and copy this entire library in a matter of hours. The study of nucleic acids has moved far beyond the classroom and into the very center of our modern world. Our understanding of the genetic code has given us the power to identify the tiny, unique variations that make us individuals. This is the basis of DNA profiling, because every person, except for identical twins, has a slightly different sequence of junk DNA between their genes. We can use these differences to solve crimes, identify ancestors, or determine paternity with near certainty. It is a biological fingerprint that can never be wiped away. In medicine, we are entering the era of genomics. We can now sequence a person's entire genome to identify glitches in the code that might lead to disease. This allows for personalized medicine, where treatments are designed specifically for your own genetic makeup. For example, some cancer treatments are now chosen based on the specific mutations found in the tumor's DNA, rather than just where the cancer is located. We are learning to fix the software of life to heal the hardware of the body. The power of RNA has also become a household topic through the development of mRNA vaccines. Instead of injecting a piece of a virus into the body, these vaccines provide the body's cells with a temporary instruction manual, the mRNA, to build a harmless piece of the virus's protein. Your own cells then produce the protein, and your immune system learns to recognize and fight it. It is a clean, efficient way to teach the body how to defend itself, using the very same language the cell uses every day to build its own proteins. Even the food we eat is tied to this study. Through genetic engineering, scientists can move specific genes from one organism to another. Perhaps a gene that allows a tomato to resist frost, or a rice plant to produce more vitamins. While this technology brings up important ethical and environmental questions, it is a testament to the fact that the language of nucleic acids is universal. A gene from a bacteria can be read by a plant because the code A, T, C, and G is the same for almost every living thing on Earth. This universal genetic code is the most powerful evidence we have for the common ancestry of all life. So you see, nucleic acids are more than just long chains of atoms. They are the history books of our species, the instruction manuals for our bodies, and the bridge to our future. They are the silent, steady heartbeat of biological time. We have now explored the archive, the stable double helical library of DNA, and the busy, single-stranded messengers of RNA. We've seen how 4 simple nitrogenous bases can store the infinite complexity of a human life. We have understood how the rungs of the ladder provide the mechanism for inheritance, and how the working copies of RNA bring those instructions to life. But instructions and workers are not enough. A factory needs a constant supply of power to keep the machines running and the lights on. Even the act of copying DNA or building a protein requires a massive amount of energy. Where does this energy come from? How does a cell take the raw energy from the sun or from food and turn it into the refined energy it can actually spend? Let's hold that thought here. A complete piece of the puzzle. We have the blueprints. Now we must look at the power plant. Our next lesson will take us into the world of chemical work and biological fuel. When we return, we will begin our study of Introduction to Metabolism and Energy. Rest your mind now. Visualize those long, elegant spirals of DNA tucked safely within you, holding the wisdom of 4 billion years of evolution. Your body is a vessel of ancient coded information, quiet and secure. There is no more to do tonight but rest, and let your cells perform their silent maintenance. Lesson 9. Introduction to Metabolism and Energy. As we have journeyed through the microscopic corridors of the cell, we have admired the architecture of the molecules that make life possible. We have marveled at the blueprints of DNA, the machinery of proteins, and the boundaries of lipids. But even the most sophisticated factory, filled with the most talented workers and the most precise instructions, will remain silent and still without a source of power. A cell is not a static object. It is a shimmering, constant flux of activity. To maintain its order, to grow, to repair, and to move, it must master the flow of energy. Tonight, we explore the heartbeat of cellular existence, metabolism. This is the sum of every chemical reaction that takes place within you at this very moment. It is the process of taking the chaotic energy of the outside world and refining it into the precise, usable power that allows a heart to beat or a thought to form. We are going to look at how life balances the act of building up and breaking down, all while obeying the ancient, unbreakable laws of the universe. As we delve into the world of biological energy, I want you to imagine your body as a perfectly tuned engine, humming with the quiet energy of 1,000 chemical fires. Let the steady rhythm of this metabolic cycle lull you into a state of profound rest. To understand metabolism, we must first view it as a vast web of interconnected pathways. Rather than a single sudden explosion of activity, the cell manages its chemistry through a series of small, controlled steps. We call these metabolic pathways. Each step in a pathway is like a single worker on an assembly line, slightly altering a molecule before passing it on to the next. This methodical approach allows the cell to manage its resources with incredible efficiency. We generally divide this vast web into two primary directions, catabolic and anabolic. You might think of these as the demolition and construction crews of the cell. Catabolic pathways are those that break down complex molecules into simpler ones. When you eat a meal, your body begins catabolic processes to dismantle the proteins, fats, and sugars into their basic building blocks. This process is essentially downhill in terms of energy. As the large molecules are broken apart, they release the energy that was stored in their chemical bonds. Conversely, anabolic pathways are the uphill processes. These are the reactions that take those simple building blocks and use them to build the complex structures of the cell, such as new muscle fibers or a fresh strand of DNA. Because these reactions are building order out of chaos, they require an investment of energy. They do not happen on their own. In the economy of the cell, the energy released by the downhill catabolic reactions is used to pay for the uphill anabolic ones. This fundamental concept is known as energy coupling. But energy is a slippery thing. It cannot be held in a bucket or stored on a shelf like a physical tool. The cell needs a way to capture the energy released by catabolism and transport it to the site of anabolism. This is where we meet one of the most important molecules in all of biology, ATP, or adenosine triphosphate. If glucose is the gold bullion stored in the vault, then ATP is the cash in your pocket. It is the immediate universal energy currency of life. An ATP molecule consists of an organic molecule called adenosine, attached to a string of 3 phosphate groups. These 3 phosphates are like a tightly coiled spring. They are all negatively charged, so they naturally repel one another, yet they are held together by chemical bonds. Because they are unhappy being crowded together, the bond holding the 3rd phosphate is very easy to break. When the cell needs energy to perform work, it snips off that 3rd phosphate, turning ATP into ADP, adenosine diphosphate. This simple act releases a burst of energy that the cell can use to move a muscle, pump a molecule across a membrane, or fuel a chemical reaction. The cell then uses the energy from food to recharge the ADP back into ATP, creating a continuous, circular flow of power. If your mind is still following this energetic flow, let us consider the rules that govern this entire process. Biology does not exist in a vacuum. It must follow the same physical laws as the stars and the oceans. These are the laws of thermodynamics. The first law, often called the law of conservation of energy, tells us that energy cannot be created or destroyed. It can only be transformed from one form to another. When a plant captures sunlight, it isn't making energy. It is transforming light energy into the chemical energy stored in the bonds of sugar. When you walk, you are transforming that chemical energy into the kinetic energy of movement. The second law of thermodynamics is perhaps more profound and a bit more sobering. It states that every energy transfer or transformation increases the entropy or disorder of the universe. In every reaction, some energy is inevitably lost as heat. This heat is a disorganized form of energy that the cell cannot use to do work. This means that no process is 100% efficient. As you rest tonight, your body is constantly radiating heat into the air around you, a byproduct of the millions of chemical reactions keeping you alive. Life is a remarkable defiance of this law, at least locally. While the universe as a whole is moving toward a state of maximum disorder, living things are islands of incredible order. We maintain this order by constantly taking in high-energy molecules from our environment and releasing lower-energy molecules and heat back into the universe. We are like a sand castle on a beach that is being constantly rebuilt by a tireless, invisible hand as the tide tries to wash it away. The moment we stop taking in energy to perform this maintenance, entropy wins, and the structures of life begin to dissolve. Historically, our understanding of this vital heat has changed dramatically. Ancient thinkers believed that life possessed a special spark or breath that was separate from the physical world. It wasn't until the 18th century, through the work of Antoine Lavoisier, that we realized respiration, the way we breathe and burn energy, is essentially a slow, controlled form of combustion. Lavoisier famously compared a living animal to a burning candle, noting that both consume oxygen and release carbon dioxide and heat. This was the birth of modern metabolic study, stripping away the mystery and revealing the beautiful, logical chemistry beneath. We should also reflect on the concept of Gibbs free energy, named after the scientist Josiah Willard Gibbs. Free energy is the portion of a system's energy that can actually perform work when temperature and pressure are uniform, as they are in a living cell. Chemical reactions are either exergonic, meaning they release free energy and happen spontaneously, or endergonic, meaning they require an input of free energy to proceed. By coupling an exergonic reaction, like the breakdown of ATP, with an endergonic one, like the synthesis of a protein, the cell ensures that the overall process is energetically favorable. It is a masterpiece of biological accounting. The principles of metabolism and energy are not just abstract concepts for a textbook. They are the foundation of your daily experience. Every time you feel the warmth of your own skin, you are feeling the second law of thermodynamics in action. Every time you feel a sugar crash after eating a candy bar, you are experiencing the rapid depletion of your blood glucose as your cells scramble to turn it into ATP. In the world of fitness and nutrition, we talk about the basal metabolic rate, BMR. This is the minimum amount of energy your body requires just to keep its factory running while you are at complete rest, to keep your heart beating, your lungs breathing, and your cells maintaining their electrical balances. And even as you drift towards sleep, your body is spending a significant amount of ATP just to maintain the status quo. This is why a balanced diet is so important. You are not just eating for fuel to move, but for the upkeep of your entire biological structure. We also see the importance of these energy transformations in the study of toxins and medications. Many poisons, such as cyanide, work by interrupting a single step in the metabolic pathway that produces ATP. Without the ability to recharge ADP back into the cache of the cell, the body's economy collapses instantly. On the other hand, many treatments for metabolic disorders involve providing the body with the specific coupons or enzymes it needs to complete a pathway that has been broken by genetics. Consider, too, the global perspective. All the energy that powers your thoughts tonight began as sunlight striking a leaf somewhere on Earth. The plants performed the catabolic and anabolic work of photosynthesis to create sugars. You, or the animals you eat, then perform the catabolic work of digestion and respiration to harvest that energy. We are all part of a single planetary energy web, a giant circuit of electrons being pushed uphill by the sun and flowing downhill through the bodies of every living thing. So you see, metabolism is the very definition of being alive. It is the constant, quiet effort required to stay organized in a universe that wants to be disorganized. It is the rhythmic spending and saving of the molecular currency of ATP. You are a shimmering flame of organized energy, held steady by the laws of physics and the elegance of chemistry. We have now explored the power plant of the cell. We've seen how life manages its economy through the coupled reactions of catabolism and anabolism, and how it uses the versatile molecule of ATP to carry energy exactly where it is needed. We have understood that life is a constant struggle against entropy, a struggle powered by the food we eat and the oxygen we breathe. But as we mentioned, these chemical reactions do not happen instantly on their own. If you were to put a spoonful of sugar in a glass of water, it would eventually break down, but it might take years. A cell doesn't have years. It needs that energy in milliseconds. To make the impossible happen at the speed of life, the cell requires a group of specialized workers we introduced briefly before, the facilitators who lower the barriers and speed up the clock. Let's hold that thought here, a complete piece of the puzzle. We have the fuel. Now, we must look at the accelerators. Our next lesson will take us into the world of biological catalysts, where we will see how the cell directs its metabolic traffic with incredible precision. When we return, we will conclude our course with the study of enzymes and catalysis. Rest your mind now. Visualize the quiet, steady hum of your own metabolism, 1,000 small fires burning within you, keeping you warm and keeping you whole. Your body is a masterpiece of energy management, tireless and wise. There is no more to do tonight but surrender to the rest. Lesson 10, Enzymes and Catalysis. We have reached the final chapter of our shared exploration into the chemical foundations of life. We have moved from the humble atom to the complex double helix of DNA. And in our last session, we looked at the vast energetic landscape of metabolism. We saw that life is a constant uphill battle against disorder, powered by the molecular currency of ATP. However, there is a final, vital mystery to solve. If you were to place a spoonful of sugar on your table and wait for it to break down and release its energy, you would be waiting for centuries. The chemical bonds within that sugar are stable. They do not want to break. Yet, your body can dismantle that same sugar in a fraction of a second to power a thought or a heartbeat. Tonight, we explore the master facilitators who make this possible, the enzymes. If metabolism is the economy of the cell, then enzymes are the highly skilled workers and precision machines that allow the assembly lines to run at lightning speed. Without them, the chemistry of life would be so slow that it would essentially stop. Enzymes are the directors of the cellular symphony, ensuring that every reaction happens at the right time, in the right place, and with the right amount of energy. As we conclude our journey, I want you to imagine these molecules as gentle, invisible hands, reaching into the chaos of the cell to bring atoms together or pull them apart. Let the logic of this biological acceleration guide you into your final, deepest state of rest. To understand an enzyme, we must first return to the idea of the energy hump. In chemistry, almost every reaction, even one that releases energy, requires an initial investment of energy to get started. We call this the activation energy. You might picture it as a large boulder sitting at the top of a hill. The boulder has a great deal of potential energy, and it wants to roll down the other side, but it is stuck in a small dip. To get it moving, you first have to give it a little push to get it out of that dip and over the edge. In the world of molecules, that push usually comes in the form of heat, which makes molecules move faster and collide harder. However, a living cell cannot simply turn up the heat to start its reactions. High temperatures would denature its delicate proteins and melt its membranes. This is the problem that enzymes solve. An enzyme is a biological catalyst, a substance that speeds up a chemical reaction without being consumed by it. It does this not by adding more energy to the system, but by lowering the activation energy barrier. It makes the dip shallower, so the boulder can start rolling with almost no push at all. Nearly all enzymes are proteins, and their power comes entirely from their three-dimensional shape. Every enzyme has a very specific pocket or groove on its surface called the active site. This site is shaped exactly to fit a specific molecule, which we call the substrate. Imagine a lock and a key. Only the correct key has the right shape to fit into the lock and turn the mechanism. When the substrate wanders into the active site, the enzyme undergoes a slight change in shape, snuggling around the molecule in what we call an induced fit. It is like a purposeful handshake. Once the substrate is tucked into the active site, the magic of catalysis happens. The enzyme might hold two molecules in the perfect orientation so they can bond, or it might place physical stress on a bond, stretching it until it snaps. It might even create a tiny specialized microenvironment, perhaps slightly more acidic or basic than the rest of the cell, that favors the reaction. Once the reaction is complete, the products are released, and the enzyme returns to its original shape, perfectly unchanged and ready to grab the next substrate. A single enzyme molecule can perform this cycle thousands of times every second, acting as a tireless, microscopic machine. If your mind is still drifting along with the motion of these molecular machines, let us look at the subtle ways the cell controls its workforce. Because enzymes are so powerful, the cell must be able to turn them on and off with extreme precision. If every enzyme in your body were running at full speed all the time, your metabolism would spiral into chaos. The cell manages this through a process called regulation. One common way to control an enzyme is through inhibition. There are competitive inhibitors that act like a broken key stuck in a lock. They mimic the substrate and block the active site, so the real substrate can't get in. Then there are non-competitive inhibitors that bind to a different part of the enzyme, causing the whole protein to shift its shape so the active site no longer works. It is as if someone leaned against the back of the lock, causing the keyhole to warp. The most elegant form of control, however, is feedback inhibition. Imagine an assembly line making a specific amino acid. When the cell has enough of that amino acid, the end product travels back to the very first enzyme in the line and binds to it, shutting it down. This tells the factory to stop production because the warehouse is full. When the levels of the amino acid drop, the inhibitor falls off, and the line starts moving again. It is a perfect self-regulating loop of biological supply and demand. Historically, the discovery of enzymes was a turning point in our understanding of the nature of life. In the late 19th century, the chemist Eduard Buchner discovered that yeast extracts could ferment sugar into alcohol, even if no living yeast cells were present. This proved that the vital force of fermentation wasn't a magical property of the living cell itself, but was carried out by specific chemical substances, which he called enzymes. This work won him the Nobel Prize and bridged the gap between the study of biology and the study of pure chemistry. Later, in the 1960s, Daniel Koshland refined our understanding with the induced fit model. He realized that enzymes aren't rigid locks, but flexible structures that change shape to facilitate a reaction. This flexibility is what makes them so efficient. We also discovered that many enzymes cannot work alone. They require helpers, called cofactors, like iron or magnesium, or coenzymes, which are often the vitamins you take. Without these tiny helpers, the enzyme is like a power tool without a battery. It has the shape of a tool, but it lacks the power to do the work. This is the fundamental reason why vitamins are essential for your health. They are the spark plugs for your metabolic engines. The presence of enzymes is felt in every moment of your physical existence. You don't have to think about digesting your dinner because your saliva, stomach, and intestines are already flooded with enzymes like amylase, pepsin, and lipase. They are dismantling the complex carbohydrates, proteins, and fats into the simple building blocks we've studied in previous lessons. When you feel a second wind during exercise, it is because your enzymes have shifted gears, accelerating the breakdown of glycogen to flood your system with ATP. We see the power of enzymes in the world of technology as well. If you use a laundry detergent that claims to remove tough grass stains, you are using the power of proteases, enzymes that eat away at the proteins in the grass. When if you enjoy a piece of lactose-free cheese, you are benefiting from a process where the enzyme lactase was used to pre-digest the milk sugar. We are learning to harness these biological machines to perform industrial tasks with far less energy and waste than traditional chemical methods. In medicine, enzymes are both the targets and the tools of healing. Many of the most common medications we use are actually enzyme inhibitors. Aspirin, for example, works by inhibiting an enzyme that produces the chemical signals for pain and inflammation. Many life-saving HIV treatments work by blocking the enzymes the virus uses to copy its genetic code. By understanding the exact lock and key of an enzyme, we can design keys that stop disease in its tracks without harming the rest of the body. Furthermore, the specificity of enzymes is what allows for the incredible diversity of life. Every species has a slightly different set of enzymes, allowing some bacteria to eat oil spills, some plants to produce colorful pigments, and humans to build complex brains. We are the sum of our enzymatic reactions. Even the aging process and the way we respond to stress are tied to the health and efficiency of these molecular workers. They are the true guardians of our biological integrity, working tirelessly from the moment of conception until the very end. Take a moment to let that idea rest in your mind. You are not a static object. You are a continuous roaring river of chemical reactions, all held in a perfect, quiet balance by these tiny, folded ribbons of protein. They are the reason you can breathe, the reason you can dream, and the reason you can recover. We have reached the end of our journey together through the chemical basis of life. We began with the tiny buzzing atom and the alphabet of the elements. We looked at the bonds that tie us together and the water that cradles us. We explored the scaffolding of carbon and the four great macromolecules, the sugars, the fats, the proteins, and the codes of DNA and RNA. We saw how energy flows through the cell and how enzymes accelerate that flow to the speed of life. You now possess the foundational knowledge to understand the world at its most fundamental level. You know that life is not a mystery of vital forces, but a masterpiece of chemical architecture and energetic logic. As you move forward, whether into deeper study or simply through your daily life, you can look at a tree, a meal, and or even your own reflection, and see the invisible dance of the molecules that makes it all possible. Let's hold this entire picture here, a complete puzzle, every piece in its place. You have traveled from the what of atoms to the how of enzymes. You have seen the logic of the universe reflected in the biology of your own body. There is nothing more to learn tonight. Your journey is complete, and your mind is full of the quiet wisdom of the molecules. Imagine your enzymes now slowing their pace as you drift deeper into sleep, maintaining the steady rhythmic heartbeat of your rest. Your body knows what to do with this knowledge. It is already using it to build and repair the masterpiece that is you. Sleep deeply now. The world is steady. The chemistry is balanced, and you are exactly where you need to be. Good night.